So I've talked to you about how the nervous system is required to activate contraction of skeletal muscle cells. This occurs by the axons being depolarized and depolarizing the axon terminal, which then causes calcium to come in, which then causes vesicles to exocytose their contents, which is acetylcholine, and that acetylcholine binds the receptors on the sarcolemma on the postsynaptic side. These then open up channels that allow sodium to come in and cause a depolarization or a more positive membrane potential on the skeletal muscle membrane. All of this is essentially electrical excitability. And so we're going to take some time here to talk about how the nervous system cells, known as neurons, as well as the muscle cells, are electrically excitable. When we move ions across a membrane, we are changing the charge that they carry. And this is what electrically excitable means. So the membranes of both neurons and muscle fiber cells exhibit voltage changes. Here it says in response to stimulation. The stimulation in this case for a muscle cell is that binding of acetylcholine to the receptor, to the channel, the ligand-gated channel. When we talk about this electrical activity or this change in membrane potential, it's called electrophysiology when we're looking at this electrical activity of cells. This activity is electrical because it has to deal with the movement of those ions. And remember, ions have these charges on them. The entire reason why we've had to review ions and what a cation is and an anion is to really understand the physiology of these excitable properties. And so there's a difference in ion concentrations in the intracellular fluid, so what's inside the cell, versus the extracellular fluid, what's outside the cell. And when you have a difference like that, that creates a voltage change or a membrane potential. So what I want to do is I'm taking an imaginary cell here, and instead of drawing one, I've provided a picture. This cell is empty right now, but it has the phospholipid bilayer on it. You can see that. The intracellular fluid, or the ICF, has a high concentration of negative ions, and we call these anions. Okay, and these are negative. These anions are going to include things like proteins. If you recall, the nucleus contains DNA. The DNA undergoes transcription to RNA. RNA then undergoes translation to produce proteins. Well, proteins have a negative charge, as well as do many of the organelles and the proteins that make up these organelles. Well, the organelles in these proteins cannot just cross this membrane. If you recall, that's the purpose, or one of the purposes of the cell membrane, is to control what enters and exits the cell membrane. And so all of these components within the cell that cannot leave a net negative charge compared to the extracellular fluid, which is positive. So you have more positive charges or a more positive extracellular environment than you do negative. And that's just the basic cell in an extracellular environment. So both muscle cells and nervous system cells respond to two specific ions, potassium and sodium. And these are the ones that, for the purposes of this course and for understanding how electrically excitable cells work, we're going to talk about. Potassium ions are higher inside the cell than outside the cell. And you can see here I've put in several potassium ions inside the cell and only 3 potassium ions outside the cell. You can draw those in. I recommend you drawing them in if you are writing on your PowerPoint slide that accompanies this lecture. Then on the flip side, another positive ion, our sodium ion, There are way fewer sodium ions inside the cell than there are outside. And if you recall, based on our differences of ions across semipermeable membranes, this creates a drive, okay? So in this case, our potassium ions, there's more inside than there are outside, and so potassium wants to go which way? Hopefully you said out. Potassium wants to leave the cell. Remember, molecules want to be in equal concentration. So potassium wants to even itself out. There's more of them inside the cell than outside, and so they want to leave. So the concentration gradient, or the driving force, is making potassium want to leave. But this is a semipermeable membrane. They can't just walk out the door. Now, on the other side of this, we've got sodium. Sodium has a higher concentration of molecules outside the cell than inside. There's only 2 inside the cell in this example, and there are many outside the cell. So, which direction do you think the sodium molecules want to go? Inside or outside the cell? They want to go inside because they also want to go down their concentration gradient. This difference in concentration of these particular ions, in addition to this negative charge that is held by the anions inside the cell, are going to determine the membrane potential, what we call the membrane potential, or the overall charge difference across the membrane potential. We abbreviate it with a VM. So again, the difference in this charge between those 2 points across that membrane is called an electrical potential or voltage. We'll really refer to it a lot as a membrane potential. In this particular instance that I have drawn here, the membrane potential is negative because it is more negative inside the cell because of these big anions that cannot leave than it is outside the cell. All right, so our membrane potential at rest, when nothing is changing, nothing's allowed to move, no sodium, no potassium are moving, is a net negative charge. And we call this, again, a resting membrane potential. This also can be considered a non-stimulated cell. So I drew here these arrows to represent the driving force of these ions. But when we're talking about resting membrane potential, there is no movement because the cell is not stimulated to allow ions to move. So at resting membrane potential, we have no stimulation, no movement of ions, sodium or potassium. And so our resting membrane potential is net negative because of these negative anions. So the resting membrane potential is usually around negative 65 millivolts, just to give you an idea and a starting point.